So I'll start. So it has been a little bit of a crazy weekend. I just got back from a wedding. So I just want to let everybody know the wedding was in Denver, Colorado. And just to be fair, it wasn't my wedding. Otherwise,
You got to speak up.
oh, really? You can't hear me?
Yeah, either one of the two.
Okay. I'll try to speak louder. I think it'll be. Uh,
You go ahead. You just talk. I'll play.
You'll play? Okay. So anyway, it wasn't my wedding, and I'm getting a little nervous because I'm headed to that 50 years. So I'm not saying in competition with anybody here, but since we're going to be talking about aged diseases, Alzheimer's and Parkinson's, so I feel, going up to 50 years is appropriate to start off this conversation. So I just want to actually give you a backdrop of where we are and what we're doing. There'll be people on the line as well. This is very unusual that we're having a collaboration that obviously the medical center and the agricultural community and the farming community in general works together, but it's almost always one-sided. You go and you see a doctor and they give you a prescription, they take a lot of money, but I think in this case, this is a true collaboration. So we're there and we're working together with the farming community. It's not just us. It's meaning us at the medical center. It's the group that's really doing the science and moving things forward. So we did speak here a bunch of years ago when we started out, and we've come really a long way. So we're going to do a couple things this time. We're going to talk about what product we have. So you're going to learn about the science and the technology, and we'll try to make it to the point where everyone will understand it. I may not understand it. I'm just talking about it. I got people in the lab to help me out. So we'll talk about the science. We'll talk about the relationships we have, the Oaklings. I think this has been really cool. We've been together for many years. I think it's close to 4 or 5 years already. So we've been friends, collaborators. We even shared a publication together. So we've done a lot of things together, and it's really beyond just the science and the technology. And we started a business together. And Joel Albertson is one of our partners, senior partner, the chief business officer. He's in the back here. He's going to be asking questions and interviewing you at the end of this, just to make sure that you understood what we talked about. And I just want to say that there will be an examination at the end of this. So you have to score at least an 85% on the exam, otherwise no food. I just want to say. So that's really an incentive for you to pay attention to what is actually going on. Oh, you think I'm kidding? I'm not. You're gonna so all true as we move forward. But there will be some questions, and the reason we're going to do this, it's not the examination of the pass-fail. It's the interest in developing a product in different parts and applications of our state, because it is unique, because very few, or any, have done this kind of thing before. We really want to engage all of you together as we move forward and build this enterprise, which is a company called NeuroRegen. So let me give you a background, then I'll go into the slides. I'm not going to be speaking for an hour. It'll be about 20 or 30 minutes, and then we'll open it up for questions back and forth and things of what you think, do, support. There's a whole bunch of things that can go afterwards. But the idea really centers around two very bad and difficult diseases, right? One is Parkinson's, and the other is Alzheimer's disease. So these are degenerative diseases. They relate very closely to aging or aged, so they're age-related. And fortunately or unfortunately, I think it's mostly unfortunate, these diseases are related in many ways to living in Nebraska. And it's not simply because we're exposed to herbicides and pesticides in the farming community. It's true, we live longer in Nebraska. Everybody know that people who live in Nebraska live longer?
Gotta hold that up.
Okay.
There's some people having a hard time hearing that.
Okay, I'll make it even easier. I'll put it right up to make it easily said. But people live longer in Nebraska, actually. And when you compare the actual age of those who are here versus New York, it's almost 6 years. So it's a big jump. It's a healthy living, the good life, but people do live longer. And these diseases are associated with aging. There's no question about that. There's genetics, and we'll talk about that in a minute, but there's that link. So we all see it, do it, experience it, or know people. Just for the sake of engagement here, how many in this audience know someone, whether they can be close, a friend, relative, that's struggling with Parkinson's or Alzheimer's disease? Okay, so almost everybody rose their hand. So obviously these are a timely, common engagement, very, very important process. So let me go. So the NeuroRetail is a company. It's just a young company. It's a biotech that really started as part of this relationship. So what I'm going to try to do in the best way I can is explain the history of the company, what it's doing, why it's doing it, and then, as I said, have some engagement questions for people who are interested in what we're doing. But this is a Nebraska company. It's really built from this community right here. And we're trying to find really some important answers to the diseases that you all kind of raised your hand about. So it's timely, it's important, it's linked to these disorders, and it's very much important to what is actually ongoing in this community and communities all over the world. So it's important. You want to push the, okay. So I just want to see that the problem is enormous. I just said that, but I'll say it again in a different way. Alzheimer's and Parkinson's in the United States, there's 7.4 million Americans at 65 and over are living with Alzheimer's today. This is really huge numbers. The projected numbers by 2060, in the years ahead, the decades ahead, we're talking 13.8 million. This is enormous. It's an enormous part. The annual cost of Alzheimer's care, it's not simply you have a disease, you go to a doctor, strep throat, or even a cancer that can be treated. You go, you see, sometimes you're hospitalized, you get tests, therapies are given, and you come back. But this is a really familial disease because we're all clearly engaged, clearly affected. The family, the community, it's really more than an individual. It's all of us who know or are engaged in the disease itself. So we're talking about $409 billion in annual costs. So it is absolutely enormous and is going to be even more enormous as the years and the decades move forward. The annual economic burden for Parkinson's now, that Alzheimer's is obviously more common, is also in the billions, nearly 100 billion, 82 billion. Next slide. Okay, so why are there failures? I mean, we have antibiotics, right? We have anti-cancer drugs, we have vaccines, and we have a whole cadre of medicines that can treat so many different things. So what's the problem? Why haven't we found, I mean, obviously it's important, the numbers are absolutely huge, the engagement is huge, the effect of the community is huge. So why have we failed? Why haven't we really have something that will make a significant difference in this disease complex, either the onset, the diagnosis, or the epidemiology? Well, it's parcel of what we call complexity, meaning normal proteins that we have in our brain, even in our periphery, become abnormal. So the first thing that happens is that during the aged process and the right genetics and the environment, and we'll get into that in a minute, but there's a number of factors. There's not one. There's no gene that says, yes, you're going to develop Alzheimer's. There is genes that are associated with Alzheimer's and Parkinson's, but there's not a direct cause relationship. It's really much more complex in terms of these interactions between the environment, the genetics, the inflammatory, and the aged process. So the first thing that happens is normal proteins become abnormal. And what do we mean by abnormal? Well, there's proteins in the brain that are ahead, and there is actually a constipation of the inability of the brain or the brain cells and the environment to deal with this change in how these proteins, these normal proteins, become processed. So a normal protein starts to fold, starts to aggregate, starts to change its configuration, either nitrosylation, but they actually evolve in a way that can't be handled. So you have a protein, and normally during the course of one's life, it's handled pretty well. But at a certain point in time, in the right environment, in the right age, in the right composition, these proteins start to aggregate. They start to come together. They start to change their configuration. They change their structure. They change the way the reaction to them evolves. And they become misfolded. And they're not dealt with. They're not handled by the body, in this case the brain, and they accumulate. Eventually they become plaques or tangles, and these accumulation or the accumulated proteins in the case of Alzheimer's is something called tau or amyloid beta, but they cause inflammation, they cause degradation, they start damaging the nerve network and the communication pathways between nerve cells. And this is insidious. Insidious means it's relatively slow, but progressive over time. So inside the cell, the tau proteins, they twist, the tangles, they block the neuron or neuronal connections, the transport, and the cell functions become dysfunctional. So these nerve cells that are sending signals, they're connecting, they're communicating with different parts of the body, they're affecting our cognitive thinking, our behavioral functions, they start becoming dysfunctional. And eventually these nerve cells die. And as they die, obviously our abilities to think, react, interact, and function, as well as behavioral changes occur in parallel. So this is not simply somebody just forgetting where they park the car, the keys, or remembering or not remembering what happened the day before, or their appointments, even our communications on a day-to-day basis. It becomes a lot more complex than that because it's simply an inability or the evolution of an inability to maintain normal life events, communicate with one another, change, react, remember, and then ultimately this affects one's behavior as we get further along. So the immune system is what tips the whole thing over, because once these misfolded aggregated proteins continue and they change their configuration, they elicit an immune response. Just like any other abnormal microbial infection in the periphery, you get the swelling, you get redness, you get pain, but in the brain reacts in a little different way. This inflammatory response is a cascade. So once the inflammation starts from this misfolded, aggregated, and changed proteins that induce a localized immune response, that cascade is circular. So the more damage or the more change in these folded or misfolded proteins, the more inflammation. And the more inflammation, the more aggregation. So it's this circular event where one affects the other and one affects the other. It just goes on and on and on. And there's nothing that really stops the process. So the plaques form, the tangles form, the inflammation ensues, and then it feeds back and makes more plaques and more tangles. So the big issue is, how do you make this go away? How do you stop this process, this circular process? You have the genetics, you have the environment, you have this misfolded protein and the change, the local inflammation, and this cascade of the circular events makes people sick. And the sick, it's not, they're mentally obviously changed, but it's not that they have a specific problem that you can treat with an antibiotic, or you change the environment, or you give a vaccine, or there's a medicine that'll work. There's really very little that can change this cascade, and this circular cascade that goes on and on. So a lot of people have tried, a lot of medicines, and we'll mention that now, but we still don't have an answer, a real answer that changed the course of this disease complex. So after 30 years, we still don't have an answer. So we can treat the symptoms. You know, people are agitated, their behavioral is affected, they're belligerent, they're hard to deal with. I mean, we can give them different medicines to calm or calming. We can have medicines that have modest efficacy, and they've been touted as something super important. I mean, you've heard about this in early disease and interventions with these antibodies, but they're just not that good. I mean, they do work in a certain subpopulation or sub numbers of patients, but they don't change the course. And they're ameliorative, but they're not significant in terms of halting the disease complex. They work, yes, that's true, but they also have side effects. So it's not just 100% positive. Could we go back to that slide? I'm sorry. So why? Because they have side effects. And the early amyloid vaccines caused meningoencephalitis. There's a real problem in terms of toxicities that occur. There's edema in the brain, not in the periphery, that can alter the course of this process and make people actually quite ill as a consequence of the therapy. And the targeting, and most antibodies actually bind broadly rather than have specific conformation. So amyloid this is one of the big target proteins that we try to affect. But it's, in the case, is how do we differentiate something that's normal from something that's abnormal? And how does the antibody make that recognition state? And that non-recognition, so to speak, or non-specificity is what causes other problems down the road. So yeah, we have drugs, we have antibodies, we can intervene, but all of them have secondary toxicities. They don't affect the course of this disease for everyone, just a small segment of the population is treated. And there is a big, big problem because the disease will continue. It's not ameliorated. It's slowed and almost in a specific instance. Next slide. Okay, so what we're going to talk about is how do we put together two technologies that could lead to one therapy. So I'm going to give you the bottom line. This is what we're trying to develop in this company. And we believe in the work that we're doing and the support and the preliminary data that we're accumulating, mostly in animals. We haven't yet moved this to humans in any significant way. We've done a few early phase clinical trials in Parkinson's in the same theory, but with a different type of approach. So we're going to talk about fucosylated colostrum extracellular vesicles as a platform for conformation together with specific antibodies. This is kind of where we want to go. The pieces are still coming together. We're not there yet. We have some proof of concept, and we're going to talk about really what we want to do and why the farming community, John and Carrie has been, and family, have really been part of this growth, enterprise, and collaborations. It's more than the parties, by the way. Okay, everybody hear me all right? Nobody hear me, okay. Okay. I think nobody is, I think of my past life, which is a lot of years because I'm an old guy, nobody's really complained about me not talking enough, or mostly it's too much. So this is the first time. I'll talk a little bit louder as best I can, as I can. So let's start. So that's the background. Johnny, you awake?
Yeah.
Okay.
Wait for you to go.
Okay, so that's over. I'll see you tomorrow. Nice talking to you guys. Yeah, it won't blow up if we go. So we're going to be tackling the problem with this new platform. And again, we're going to be using Alzheimer's and Parkinson's almost interchangeably, so you'll have to follow me a little bit down. So when we talk about the protein that's chemically damaged, we went through this. Alpha-synuclein is that misfolded protein in Parkinson's, amyloid beta and tau in Alzheimer's disease. This is a distinct, a targetable epitope, so we can go after this misfolding process, this abnormal accumulation of these proteins that actually are causal in this disease complex. The microglia is the cells in the brain that we talked that are responsible for the immunology, or the immune responses, the inflammation, and the secondary damage. We have blood cells that also are part and parcel of inflammation and clearance, and most of the cells that we have in our body and the periphery are used to to recognize and eliminate pathogens. That's why we have vaccines or treated to prevent disease. But we also know that if we get a splinter or a cut or a scrape that we have, there's an inflammatory response, there's redness, swelling, and then ultimately resolution. There's similar processes that go in the brain, but again, it's not the same, but there is inflammation. That's the linkage. And the cell that causes that inflammation is called a microglia. It's a small immune cell that reacts to the damaged proteins and produces inflammation, and that inflammation ultimately damages nerve cells. This cascade that I talked about just before, between the misfolded proteins, the inflammation, the activation, leads to the damage or ultimate death of the thinking cells of the nervous system. And in the case of Parkinson's disease, it's a dopamine neuron. In the case of Alzheimer's, it's some of the parts of the brain we call the cortical neurons that are more affected. But the nerve cell is the one that ultimately dies as a consequence of this disease complex. Next slide. Okay, so what do we do? So what are we trying to do here, and what am I trying to talk about? Well, we're trying to develop two independent pathways that ultimately will come together. And there are people in the audience, my scientists, you turn around, are working on these pathways independently and together. But I'm going to give you the history of where we were and why we picked these two independent pathways. So this is your first question on your examination after this is over. What are the two pathways? Well, one has to do with inflammation, right? We have to control the inflammatory response, right? That's absolutely critical. And the other part has to do with somehow preserving these nerve cells that are dying and sick as a consequence of this disease complex. So we're knocking out the inflammation, we're trying to control the inflammation, and at the same time, we're preventing the progression, or what we call the degenerative pathways. So it's an inflammation and protection at the same time, working hand in hand. So what does this have to do with farming. Where does that go with us? Well, we have a humoral arm. We're targeting the antibody, which basically is where we started, and the regulatory cells are engineered to actually dampen the inflammation. So how do we put the pieces together to generate a therapy, a new therapy, that could really, we hope, will revolutionize this disease, or the therapeutic parts of this disease, the disease complex? How does that happen? What do you think? Did I make any sense? A little bit. No, I was just dancing this morning. I mean, I was at a wedding, you know what I mean? I wasn't talking about this stuff. I was just drinking a little beer, but so if I don't make any sense, it's just a little bit of a hangover from this morning. It's kind of a problem. Okay, so the idea is to clear the toxic proteins that have been aggregated and calm the inflammation and put it together into a single product. So next slide. Okay, so this is kind of what we're trying to do, and it's really complicated. So I'm going to make it simple. You're laughing. I'm going to make you draw this at the end of this. I'm going to take this over. How am I doing? Am I doing okay?
Doing good.
Okay. So let's take a minute here, if we can, and try to explain that. So at the top of this, do you have a pointer? We can point to the top where is a picture of a halfway of a brain that's cut down in the middle. It's that pink and red thing in the upper left hand and upper right hand corner. One's is a sick brain and the other is a healthy brain. You follow me so far? Everybody knows the difference between a sick brain and a healthy brain? Yeah. I've had that problem too, because I have what you call a dean and a president of the university, so I'm to differentiate the two as best I can. Well, you're laughing. I have to live with this. You can laugh about it, but I have to live with it. So what happens is, I told you, right below it, there is what we call the alpha-syn, the monomer, the oligomer, the protofibril, all these fancy names, which it aggregates into what we call a Lewy body in the case of Parkinson's disease. So that represents this misfolded proteins. It starts as a single protein, right? And then it comes together in different configurations in the environment. It changes what it is and its structure. And that is the one that incites the disease. Ultimately leads to what we see in the upper left-hand corner. And what we're trying to do is control it to take that degenerating brain, you can see in the upper left-hand corner, and make it into something that's recovering, a recovery brain. So that's why you see the person that's kind of hunched over with Parkinson's disease. Could be Alzheimer's, but it's Parkinson's disease with the movement, the tremor, the ambulation, the gait disturbances. I mean, it's all part of that disease complex. And somehow we want to have an improved outcome. That's what we want to do. So all the stuff in the middle, where we talk about the pro-inflammatory mediators, the clearance of the misfolded proteins, the halting of the degeneration or the degeneration of the neurons, the nerve cells, their survival, and then ultimately leading to an improved outcome. So we have two ways to do this that we're thinking in terms of our science and our technology and our tests and laboratory and animal models, is we want to affect both parts of this disease, the inflammatory component and the aggregation of these proteins that lead to nerve cell destruction. All right, so the question is, how do we get there? Next slide. Okay, so now you may recognize this. This is my Uncle Harry on the right. No, it's not. Yeah. So, okay, this is a really important exam question, and you better pass this. What is this on the right-hand side? Anybody recognize this? Okay, how many people think it's a cow? Okay, nobody thinks it's a cow. Okay, I'm telling you, if you can't recognize that this is a cow, I'll see you in my clinic and we'll do a cognitive workup and see what happens, all right? I just want to tell you, so you don't think I'm a liar, This is a cow. This is a cow. Okay, yeah, we're gonna get there. Okay, so the question is, what does a scientist who spent the last 45 years of his life working on cells and inflammation, regeneration, stem cells, development, all of a sudden, after 45 years, come back to cows? How do you think that happened?
You got smart.
Well, you got to. I recognize the cow. I mean, I was here. Do I know the difference between a cow? I'm going back to basics here. And being smart is probably the wrong answer. You got to talk to my wife because I'm not sure she would agree with that, but maybe she would on certain parts of this. But we were faced with a dilemma. Okay, let me just, and everybody understands that colostrum is a big market. I think we all understand that. It started 4 or 5 years ago, and it made about $4 or $5 million. Right now, you go to Walgreens on the shelf, and it's all over. Now the market is at $4 billion. Now I'm not exaggerating this; it's all true. So, and I'm going to say something that you're not going to believe, but you're going to, you just got out of church, so it's going to be important to say this. There hasn't been any real scientific study of looking at the cow product, colostrum, that shows that it does anything important to affect any of the functions that is being used or sold by, right? So it's making billions of dollars. There's no science behind it. It's just like, how do you feel? I feel pretty good. And they've been able to market this in a very significant way. It just shows that you can say and you can believe because somebody says it and you believe it, right? You ever hear that before?
Yeah, I just let a beer could do the same thing.
A beer?
A beer could do the same.
Yeah. Well,
Like you feel good? Yeah.
you follow me? Barely, not so much. Now, I'm not sure what's true or not true. I can bring in my old boss and ask him. Everybody knows who I used to work for before I came to Nebraska. Maybe I'll get in trouble if I mention his name, but you may have heard of him. His name is Tony Fauci. That's where I was before I came here. So you can believe everything that I'm saying. That is, the part of it is true. I was working at the NIH with him in the past. But that's not the answer to the question. The answer to the question is why? And what can we do medically to actually get to the bottom of this and to see that it will make a difference? Well, the first thing that we did, and the reason we actually came along, is to see if we can stimulate regulatory T cells. A certain type of T cell in the blood is a lymphocyte. We have white cells and red cells, you know about that, and platelets. I'm not going to give a dissection of hematology or anatomy, but there's blood has a number of different elements in it. And one of the elements that the blood has is a cell that can regenerate. It's a regenerative cell. It's an anti-inflammatory cell. It's a small population of the blood, but those cells actually are very potent in controlling inflammatory disease. And actually, the Nobel Prize this year was won because of that cell. It's called a regulatory T cell. And we did many clinical studies on that. We were probably, if you look in the world literature, the internet, we published more papers, did more experiments that related to this cell and the brain and regeneration, both in people and in laboratory settings than almost anyone on the planet. But we had a problem and an issue is because when we're using medicines that will induce a potent anti-inflammatory response or regulatory response, they had a number of side effects. If you modulate the immune system, especially in an elderly person, you're at risk for disease, right? You can't muck up the immune system and not see what can happen as a consequence of that. And we did clinical studies, and patients did get better, to be honest, and our careers did well, grants came in, opportunities came, but inevitably there were what we call side effects. That means it's not just positive, there's also the negative. It's kind of that balance. So we had to find something that could regulate the immune response and be part of this cascade that could be used to treat neurodegenerative disease, but do it safely. You got it? Safety is a big thing. So that's why we came and we knocked on John's door one day, and John said, "Who is this guy?" But Keith introduced me, so I guess it's all your fault. So we came to, and there's, I think there's some kind of relationship over the years, right? I think we're all related. We're like cousins at some point. Close cousins. But there is some link, I think, at some point in time. No? And we thought that it would be kind of interesting as one of the things we were testing and developing, that the elements of colostrum, where the market was so high and a lot of excitement, could actually do something good and induce an anti-inflammatory response and maybe affect these misfolded proteins that were part of this disease complex. And the people that are sitting, I won't introduce or embarrass anyone here, but I had to convince them that we were going to do this work and experiments. And some of them were young graduate students that are still involved and engaged. Do you know who that might be? Or never heard of that person? And from all over the world, that they came to be here in Nebraska and learn and develop. But we wanted to test, see what would work. And lo and behold, we did some of this work here, and we saw that in the setting of brain disease, and the work we did initially was in Parkinson's and related work in Alzheimer's, but Parkinson's was a big part, is we were able to see that they could induce these protective cells and affect an inflammatory response. And there's other things going on in the lab at that time. We were developing animal models, systems that we can actually do the testing and development. We had to figure out ways to get these cells, if they were working, into the brain, into areas of damage. And that's why the fancy word fucosylation is being developed and looked at. And it was hard to have this conference and invite all the scientists here. They're all sitting behind you or adjacent to you. But we had to give them a few hours off because they should be in the lab doing work. And they got the afternoon off. It really hurts me to give them any time off. I'm being honest, you know. You got to give me a couple of points for that, you know. I mean, what's going on? But lo and behold, the system worked. There was an anti-inflammatory response. It did build on a lot of the work that we did many years ago and up until the present time. We were very excited about the prospects of this. And we were also working in tandem to look at how we can engineer the environment, maybe not colostrum per se, but the cells that are being made as a consequence, to get to areas of damage and actually do repair. It was something very different. Nobody had done this before. And we were very excited about getting the material. And we met with John and Carrie and the family many times. We had parties and get-togethers. We were dancing at, remember that? She's quite a dancer, by the way. But it was a real relationship that we were developing over time. And that extracellular vesicles are the carrier, right? Colostrum EVs are natural, they're non-immunogenic, they're stable, they carry proteins, cargoes that are critically important that are underutilized or underdeveloped. The supply is not a constraint. So any other issue that we had to deal with in terms of finding a new protein or a new means for treatment was limited because the supply was limited. But in this case, the economics are unlike any biologic. They're autologous, they're non-immunogenic, and we can recover large amounts of this. And we can purify the active ingredient, which we believe, and based on a lot of work and work that's still ongoing, and derived from colostrum, that will affect the inflammatory cascade. We have a lot of ways to look at that, but the data was quite exciting and still exciting as we try to maneuver this into a product. So we had a stable resource. We developed friendships and collaborations and a company where we're all partners in, I guess. So far, we're partners. Okay. And they have cargoes. So the cargo it's not just the vesicles that we get from the colostrum, but what's inside the colostrum vesicles. And these are proteins and they're lipids, they're fat, they're nucleic acids and RNA that also can be used and tamed to actually induce a metabolic and a functional change in the brain and cells, and actually control the inflammation and incite a neuroprotective response that could change the disease or the disease complex. Next slide. Okay, so we spent a lot of time on this. This is a simple slide in the cartoon, and it has nothing to do with Popeye the Sailor Man or Rooted, I don't know if you're watching because of my generation, but it is a cartoon. But the issue of not just a typical cartoon is if we're working with colostrum, we can't infuse milk into a patient with Alzheimer's disease. They would probably put me away if we're trying to do that. We have to dissect the active ingredient, which is these extracellular vesicles. And then we had to do it in a safe way, a reproducible way, and in a way that we can translate whatever we do in a laboratory or an animal to inevitably to clinical trials and ultimately to treat a person who's struggling with this disease, even when we find that it has all these capabilities. But we went from this purification procedure that would take us weeks, so 24 hours plus, so it wasn't where we were trying to go or what was achievable. We had to go from weeks to hours in terms of putting this together. We had to do it quick, we had to do it reproducible, and we had to get large yields if we're going to go from a laboratory setting into a human with this disease. So centrifugation, human scale, large volume isolation, release, characterization, and purity, and then ultimately isolate the product, which I'll put right dead center. It wasn't just a vesicle that we got from milk. The vesicles contain a lot of parts, as I said, lipids and RNA and proteins that could be regenerative, could be affect this process. So we did that, and it was a lot of success. John was a co-author on that paper, the high impact paper, and others in the audience really did the work. I didn't do anything. I just kind of watched as we move forward. But we were able to actually change this process and show that it actually worked in an animal model or an animal with neurodegenerative disease. All right, next slide. Okay, but we had to build this, and what we said of producing the kick was making our own antibodies that could give us that dual collateral approach. And again, those are sitting here amongst us, actually did the work, and we're able to put this together in a collateral or a combinatorial way to dissect this. So this gave us another hit because each part alone was kind of okay, but putting the pieces together was quite significant. So we had to do the immunization, the fusing, the hybridomas and expansion, produce our own monoclonal antibodies. And we generated this because they were selected to go after these misfolded aggregated proteins that were specific for the pathological parts of this cascade. So this was, again, directed to either alpha-synuclein or amyloid beta, but putting that together, we had the capabilities to attenuate the inflammation or slow the inflammatory process, which is driving the disease. And the causal aspect of it through the antibodies, we can have a very significant 1-2 punch. And you can see where the colostrum could potentially fit into this cascade on the immunological part of this process. Next slide. But ultimately, what we want to do, I mean, with all this fancy science, we want to make it simple. So we want to use this combinatorial approach where we tackle very complicated parts of this disease complex, put this together as a treatment, and be able to inject it into a person to have a positive clinical outcome. So the idea is that, yeah, we have everything that's complicated and everything that we just talked about, but the bottom line is simplicity. It's never going to work until it's made simple. Next slide. But the medicine has to find its way, right? You ever go, we try to find our way to a place called Elmwood, Nebraska. I don't know if you ever heard of that place. We're sitting in here. We were hungry. We're looking for, I gotta finish this lecture soon because all that food is looking pretty good. But we needed a system that would be a guide, a GPS system that we have in a car, a navigational system. So one of the things that we're heavily involved in is not just developing a dual approach in getting either cells or the extracellular vesicles that we can inject and hope for the best, but we had to develop a means so these products, these medicines, will get to areas of damage. So yes, we can make whatever we can make, whether it's a cell or a vesicle, but ultimately we have to engineer that vesicle to go where it needs to go. And that's where the fucosylation comes in. And what we tried to just put this together, and this is a piece that we're working on and developing, is on the left side is CEVs, those are colostrum extracellular vesicles. And you see some of them are going across the blood-brain barrier. There is a barrier that protects the brain, and it also protects the brain for the therapeutic components that will get across to areas of damage. But we had to do what we say, engineer these cells or these vesicles to get across in higher quantities so they can elicit the kind of change, the modulation, and the clinical effects that we want them to do by modulating essentially the blood-brain barrier, the function to get them across. EVs largely stay in the circulation, but we want to get in the bottom right-hand corner this fucosylation procedure to get them into the area of injury to elicit repair. So fucose is a homing signal for sites of inflammation and used as turns a delivery problem into a targeted advantage. So now we have three parts of this puzzle. We have the anti-inflammatory part, we have the targeting of this misfolded aggregated protein that drives it, and now we have a system that we believe, and we're working on this in some earnest, that will allow us or facilitate its passage across this barrier that protects the brain, but also precludes the entry of these molecules, of these therapeutic molecules across. And if you look at the fucosylation, we're able to affect, potentially, we're doing the experiments now, that 58% of what we put in will home to areas of damage. So finding the area, eliciting the effect, and curbing the disease complex are the three parts that we're trying to put together. Did I lose you?
No,
How am I as a teacher? Am I okay?
good.
They won't let me teach in Nebraska because I tell too many jokes. Students don't want that. They want to know what's on the exam. I don't care about what the thing is. I just want to pass the exam and get their degree. It's a big problem. Next slide. Okay, so when we look at the diseases rescued, and yeah, the microphone is important. And I'm not going to go through this because it would take me another hour, and I'm going to try to fill this, but this is the work that is being done by those sitting in our audience, and they're all here today. So when I break and stop talking, you can talk to them directly, and they'll give you the real insights because they actually do the work. I'm just a conductor. I'm not playing any instrument in this part. But the idea, if you look at neuron survival, and we've done this by a lot of different means, but the idea is the stuff in the right-hand corner in the bottom. You see a lot of green neurons. Those are healthy. The red neurons are the ones that are sick. And if you see those blue circles, blobs, that's the medicine that's actually going across the blood-brain barrier into the areas of damage. And you can see the difference between the sick brain, the histology on the right, which is all brown and red. It then becomes green because we can control the nerve cell response. And we can do behavioral rescue as well. And behavioral rescue is basically showing that when we treat these animals, they're smarter. They actually can do things that they couldn't do, recognize things that they couldn't recognize, find their way. In this case, there are food pellet and recognition sites that we've actually improved their behavior when they're tested. And these blocks are the series of tests that we do over time, and the different lines that you're seeing in the left-hand part of this diagram is just showing that we can make them less, to see less errors. They actually find what they need to find. Their recognition capability is markedly improved. And the idea is the curves that are more to the bottom, the black ones, are the ones that are smarter. The ones that are brown and pink are the ones that are dumber because it takes them a long time to find what they need to find. In this case, it's food or food recognition. And we can show that these animals that are treated with anti-inflammatory based cellular therapy or amplifying the cells that are neuroprotective or treating with the antibody, that we can improve all of these parameters and make these animals smarter. And these are animals that are actually having a very significant Alzheimer's-like disease complex. Okay, next slide. So why is it so special? Dual action, and I told you about the dual action, going after the toxic proteins, calming the inflammatory response, crossing the barrier, inducing conformation that's specific, which limits what we call ARIA. These are the limitations of inflammation that's seen with some of the drugs that are on the market or were on the market. And the commodity material is that using a means to deliver this through bovine colostrum is agriculturally abundant, something that we can do and collaborate and move this product forward. So it really puts the pieces together in a very significant manner. Next slide. Okay, I'm going to be finishing up and say that this is, even though I'm telling a lot of jokes and I'm coming back from a wedding and drinking too much this morning, but I won't talk about that. I hope I made some sense, right? Followed some of the things that I said. But the idea is this is a serious operation, and I want to end with that. It's just saying that it's not that we have these grandiose ideas. We're sitting with many patents approved, talking with the FDA, moving products forward, millions of dollars that were awarded to us, both from the NIH, private donors, and something called the Michael J. Fox Foundation, who also has been heavily supportive. So yeah, I mean, people joke about things, and the university has gotten a lot of stuff, and not always good. But there's a lot of really good stuff that's going on at this university that we need to be certainly proud of. And I'm very proud of the group behind you, or to your left, where they go. We've written books. We have several textbooks that we've written on the topics. We actually have our own biomedical journal, and we published hundreds of papers, scores, and lectured throughout the world on the work that we're doing. So it's obviously a very serious operation. Next slide. So the market that just proved it exists, the regulators approved the disease, but the payers covered them and the patients accepted the risk. But what is the questions that needs to be answered? Next slide. Okay, so these are the last couple slides. People want answers. People want answers. So the idea of describing the disease or developing biomarkers or seeing a doctor, not seeing a change in terms of the clinical status and vis-a-vis Alzheimer's disease is changed. People want to see that when we're talking about $7.8 billion in the global therapeutic market, we're talking about the therapeutic market of drugs that are just minimally effective. People want change. They don't want this. They want to see that this disease is going to be adequately treated, and the symptoms and the ravages to the patient and the community and those that are close to those individuals change. So we have to really understand that the demographics are the tailwind, right? We're going to see more of this, the projected part of this. And this is a safety, aggressive move that we're working literally day and night to make this curve, or this cadre of ideas, move forward from a hypothesis or a thought to something with real change that's really going to make a difference for the patients who are suffering. That's the bottom line. Next slide. So where we are and what comes next, well, what we want to do, obviously we're advancing the science. We're working on securing funding and support from the community and from granting agencies. We want to build what we have vis-a-vis the manufacturer and our position within the community, and then scale for the impact. And for the impact is not good if I can't, nobody's hearing what I'm saying. That's a big problem. Okay, next slide. This is our final slide. So we want to partner with you guys, the therapy, and I'm here to generate interest, generate knowledge, to share our vision, and understand that those that are affected will come and communicate and engage. Ultimately, we can raise money, raise awareness, and raise interest to expand this operation and get it moving. I know it's going fast, but get it moving even faster with this kind of collaboration and cooperation, I think, together. Okay, I think that's the last slide. And just to mention the people that are benefactors, those that are actually hold the patents. I didn't mention all the scientists, some of them are here, but the people that are mentioned on the left-hand side are those that are actually in the discoverers. So they're part of the patents and part of the drugs that I mentioned. And then the benefactors, Carol and Joel and John and the family. That's really made a lot of these things possible. So thank you for your time, and let's stop for a moment and then see if we can, ask questions and have an exchange. We're going to try to have a question and answer session if we can, but more importantly, I'm just pointing to Joel Albertson. He's sitting in the back. He's going to be talking a little bit one-on-one because we're trying to get the message out. So sometimes you may not want to ask specific questions to me in a kind of a group setting, but we want to put this together in an article that will be published of what are people thinking, what are the needs, what are the concerns, what we can do to help each other, to move this problem from a problem to a solution. So I'll stop there and thank you for your time. Okay, so we'll do, why don't we take a few minute break? Can we do that? And then we'll assemble back for questions that people will ask as we move forward. And then individually, Joel, you can raise your hand so everybody knows who you are. Joel will be around to kind of talk on one-on-one to get your thoughts, questions, and answers. And I just want to mention, yes, Joel. Yes, please.
For time's sake, for everybody else, maybe because everything is fresh in everybody's mind, it makes more sense to do the Q&A now instead of reseating them again after they get up.
Okay, that's fine with me. Is that okay? Just to mention, the people who are actually doing the work, could you raise your hand? There are people, and give them a little bit of round of applause in there. Yeah, I don't want to go through everybody's name because you're going to forget their names anyway. But they're really, to be really honest, fair, those are the ones that really made things happen and made everything happen. So there's a lot of pride, a lot of appreciation, and a lot of success in seeing that their dedication and their motivation is really totally awesome. So maybe another round of applause for those people. Okay, questions? And you can ask questions to the scientists too, and then we'll break it up on an individual basis.
We've got online questions.
Online?
Yeah,
Oh, should we start with that first?
sure. Unless you have more that you want to do.
Is there anybody online or?
Yeah, there are.
Oh, they're really?
Cool. Yeah,
They're still there, or they didn't?
they're still there. They want to know, how important is early diagnosis a factor in the equation?
I'm going to give the simple answer, but I'll give the simple answer, and then we can go in, as we go into the individual level, we can get deeper into it. It's very important. The earlier we know, the better the diagnosis, the certainty of the diagnosis, the ease of intervention will be much improved. So the sooner we know, better we know, and the interventional strategies are started, the better the outcome will be. So the answer is yes.
Both.
Yeah.
Both
And Parkinson's, yeah. Yeah.
So
Yeah, go ahead.
I understand that. Do you want me to speak into that?
Yeah, please, yeah.
I understand that oftentimes when people become symptomatic with Alzheimer's and since the disease process has already been going on for 20 plus years. So I'm just curious how you plan to identify and at what point you plan to intervene. Because there are no ways to identify people until they're symptomatic right now.
Yeah, so, yeah. Well, yes and no.
Okay.
I think, let me give you the no before the yes, if I can. The plaques develop at the second and third decade of life. So I don't want to give you a bad prognostic sign, but I guarantee that if we would do a PET scan on your brain, there would be plaques. They really occur. They occur very early. The issue is not the plaques. It's what the plaques do. So not everybody that's 20 years old is demented, although a couple of my children maybe are suffering from this. I just want to say, you know how it is when you have teenage kids, you know what I mean? I don't know. Maybe it was a while ago, I don't know. Okay, we'll talk about this. But I would say the majority of people in the early decades of life have plaques, tangles, and have signs of the disease, but only a small percentage actually develop the disease. And that actually gets larger and larger as we age. So the big question is not just making a diagnosis, doing a PET scan and seeing plaques. It's really looking at the trigger. So one of the things that we're doing and that we're very interested in is what we call biomarkers. Why we can look at a person, and the biomarkers is not doing a cognitive exam and everyone's 25 years old, but actually looking at markers that are relative to what we're seeing vis-a-vis our research that will target the disease or the operative disease or the active disease, and we can get a better handle. So if a person comes in and maybe in a certain age, may have signs and symptoms in an early, but they're concerned, is this just normal aging or is this really the disease? The idea is not only the combinatorial therapy for treatment, but the combinatorial therapy for diagnosis. And we're beginning to get more aggressive because this, we're talking, the numbers of billions of people affected is enormous. And we better be able to do this earlier because the answer to that first question, our chances of successful intervention is going to be logarithmically better the earlier we can find this process. And people are not even when they have the signs and symptoms of disease, they're not necessarily going to be treated because we don't have great treatments yet. So it's the combination of the firm diagnosis, which is done by PET to SPECT and blood. I mean, there's a whole bunch of things that are being used clinically, but intervention in something that works, that actually works, and that limits the secondary toxicity. Do you follow what I'm saying, or no? Did I lose you? Oh, okay.
So is the biomarker that you're looking at, I guess, what is the biomarker that you're
Okay, well, you probably see this in the TV all the time. People are coming up with looking at amyloid or tau, developing blood tests, or looking at alpha-synuclein or changes in the, so that's a direct measurement of this misfolded aggregated protein. Most of the work that we're doing is secondary. So we're looking at inflammatory cascades, we're looking at secondary effects, we're looking at the numbers of these regulatory cells or the effector cell ratios. We're developing a whole bunch of things in conjunction with our therapeutic trials that ultimately will move us into that biomarker realm. But I think the bottom line is the same answer that I gave before. Earlier is better, and intervention with some successful candidate of a drug or combinatorial therapy. Yeah, go ahead. You want to?
If I understand correctly,
Yes.
you have already been using this on animals, mice, etc. When do you anticipate it to be something that we would go to a trial for humans?
Yeah. Well, I would love to say yesterday,
Yep.
which would be a good one. But there are strangleholds in terms of moving this forward. We have done a number of clinical trials. You can Google me and look at my name and clinical trials. We've done a bunch. Some were successful, but the issue of translation was a bottleneck. And I think the major limiting factor vis-a-vis translation was toxicity, secondary toxicity, and longevity, meaning how long these will last, because there is what we call amnestic response, which means that after we treat this patient, they develop tolerance to the treatment itself. So I think there's been a big push. There was incredible push with these early Alzheimer's antibodies. Everyone was excited. The hype was way, way above the actual events and what we saw. And then all the toxicities fold in. And people were saying, look, yeah, I'd love to take this medicine, but I'm not sure it's going to help significantly in where I am right now. So there's been this reluctance. I think the clinical trials have to have a combination of the biomarker, the early diagnosis, and the successful intervention. And what we're trying to do with Neurogen is to take a new tactic. Obviously, why do we spend so much energy on cows? Because it's safe. I don't see anybody dying of milk. Anybody ever die of milk? No. There's not a lot of death. I mean, milk is, people, you ever drink milk?
Every day.
Okay, there you go. But safety is a big issue in terms of intervention. And biomarkers are a big issue in terms of intervention. And combinatorial therapy that actually works is a big issue. And what we're trying to do at the university and beyond with this company is to put it all together. Pravin, what do you think? He's one of our lead scientists, and I think he's asleep, so I want to make sure he's awake. He's awake. He's probably just tired because you've been doing so many experiments, right?
Yeah.
You haven't been doing many experiments? I hope so. I hope so. All right.
Want me to answer what?
I want you to be yourself. Just tell me how you spent the summer. Was it good? You went to the beach, downtown. Tell us a little bit what your thoughts are in actually doing the research day in and day out in terms of what do you think? Where do you think these pieces will come together?
Something more easy going to the clinic for available people.
Can you come up, stand up?
Come here, Sandra.
And then,
See your face.
yep. And then, right into the microphone.
So what?
Can I sit next to you?
Different projects we're doing something that can go into the clinic, not necessarily clinic, but should be available in the off the Walmart or in the pharmacy section where you have the nutraceuticals. It will be the colostrum extracellular vesicles. So you can already buy colostrum powder from Costco or anywhere. So we are doing the next step up where we isolate the most important parts of colostrum, that's the extracellular vesicles, and probably make that into some kind of pill or formulation that's easily available for purchase. So we have for the other antibody therapies, and those are much more complicated because the FDA would be involved if you have to market them. So we'll have to do a lot of clinical trials. But colostrum is mostly a nutraceutical, it's a natural product. So that's something we might, if we push properly, we can get that into the market in the next 1 or 2 years. The major bottleneck I feel for colostrum is it's the volume. You need a lot of milk to get this product. So we would kind of partner with large dairy farms, and I don't know how big the local, population of cattle is. That's what I was asking a while ago. So if we don't have the numbers, we would have to go. Recently the colostrum market is also picking up, and this has become a much more precious resource. People are directly going to the farmers, and otherwise it was a base product for a long time. No one cared much for it, but that product also is going to get more and more expensive. So I think those are the two. One is the more nutraceutical focused project. The other one is the more pharmaceutical based project. So the nutraceutical one would be the easier one. So.
Yeah, and this is a debate or a discussion that we have all the time. Discuss even my dear friend, who's the chief business person and a major investor in the company, who's sitting in the back. I think how many times have we talked about nutraceuticals versus pharmaceuticals? Yeah. It's a big issue because one will be quick, and the other will be more long-lasting and have a better endpoint. So this is something that we really talk a lot about. And I think the gut, if I can say a gut is more towards the pharmaceutical than the nutraceutical because of the ultimate efficacy and the longevity of the product. But we're still working on keeping an open mind about all of this. Okay, thanks. I don't know.
Okay, we've got more questions online. We're ready.
Okay, stay here. Okay.
So this person wants to know, is this an answer to all inflammatory non-degenerative diseases? And at what point do you feel like the impact for newly diagnosed Parkinson's and those further along on the journey?
Okay, so the first, I'll take that, and maybe you'll help me. Stay here with me. The answer is maybe on the first question, because the idea of using this type of induction, I'm talking about the cellular component or the EV component, is really if we equate this with a baseball game, we're in the top of the first. And if you go and what we do is we have what's called PubMed. It's a literature search of what kind of research has been done. I'm talking about therapeutic research in Parkinson's or Alzheimer's with colostrum. And a lot of papers are on cosmetics skin disease or skin issues. The biggest use of colostrum clinically, you know what the biggest use is? Wrinkles. You know, you ever hear of wrinkles? You never heard of that? Never. I'm not trying to pinpoint anybody, but it's cosmetics in large measure. So the idea of using colostrum or components of colostrum therapeutically is that it hasn't gone yet. And why hasn't it gone? Because most of the isolated extracellular vesicles are cell-derived, so you need a lot of quantity. And the cell-derived work that was done is limited because some of these cells, when we talk about these diseases, are coming from a microphone, right? They're coming from damaged cells. So there's a yin and a yang vis-a-vis, they can cause disease itself. But I will ask Pravin, since he's standing right next to me, to circle back with the first question, that this kind of approach can be used beyond these neurodegenerative diseases. Can it have far-reaching implications for other disorders?
There is basically you can use this for any disease that's caused by anything. For example, even within our lab, we try the same colostrum EVs for epilepsy. So we have a collaboration with a different group, and they in an epilepsy model of disease in the mice compared to the normal mice which have epilepsy, you give the colostrum EV, they were able to reduce the epilepsy incidence by about tenfold or 10%.
Tenfold.
But it was significantly different. So for a nutraceutical, for a natural product, that's a big, big discovery. So the bottom line is, like I was saying, any disease with inflammation, it can be eczema, it can be IBD or any other kind. So there's a host of diseases that are just because your body is reacting weirdly to a normal process. So the basic premise is that these will induce the regulatory T cells. These are the T cells that make sure that your immune system doesn't go crazy. So the colostrum EVs promote these regulatory T cells. So we just are making sure that your body is not overreacting to things. So there's a host of diseases that we can expand into. So that's the whole point of the colostrum market. So they want to make this one shot for all kind of therapy. Not necessarily therapy, but it'll be something of a supportive system to existing big pharmaceutical drugs.
Yeah, thanks. Susmita, why don't you come up as well? Because you're the one that's actually doing some of the earlier work. But thanks. I just, we're going to wait for another question, and I'm going to ask you to be engaged as well. So stick here for a while. Another question? What are you poking your eyes out here? What's going on?
They want to know the colostrum is being used for your research. Is it being synthesized from the lab, or is all collected by bovine?
Okay. Why don't you, since you're involved in that, you can answer that question.
So the colostrum is coming from the Oberlin farm, or the active compounds.
Up to your microphone.
Microphone.
The extracellular vesicles.
Where's your
we are
husband?
deriving in
Oh,
the
there he is.
And then they want to know
Did I
is
bore
there
him
a
to death?
case where someone might be eliminated from the therapy
Oh.
I think that we do not have a yes or no answer at this point because we are at very preliminary stage. But the animal models that we have used, they had initial symptoms of Parkinson's, and they got a very good recovery with this colostrum extracellular vesicles.
Yeah, no, absolutely. And I think we're still pushing that. I think the work that Pravin and the team sitting over here, and Emma will embarrass you, and Rana and Nada, I'm not going to put everybody on the stage here, but we are exploring different kinds or different models of the same disease, whether it's Alzheimer's or Parkinson's. We spent a lot of effort on that as well. We're also looking at other diseases. I mean, we have a very big and significant paper on epilepsy. These are childhood epilepsy, obviously out. So we're doing all this. We're doing all this. But the big issue and the reason that I'm here is not to talk about mice and how mice recover, but to gain interest, support, direction in helping us and partnering with us, whether there be patients, whether it be donations, whether it be interest in the company, it doesn't make a difference. But we need to join the community together. This is a real problem. This ain't going away. And this is going to be a bigger problem in the time ahead. And either we tackle it, either we engage it, and we do it as a community together, or this will devour us. It's very serious. It's really serious. And it's going to get worse. That's just the way it is. And that's why I'm here. That's why I'm here. That's why we're here together, to gain community awareness of this. Yes?
So can you only use cow? I mean, can you use goat? Then does it just have to come from a cow?
No, first of all, the answer is no. The cows are an easier resource for us. We have them available. We have the linkage available. It works. And we have large quantities that we can use. I will mention something that was really, this is about science, and maybe it's just kind of a cool result that you guys have made. So one would think that the more of these vesicles that we add, the better the result will be. So we give a little bit, we get some result. I'm talking about protection, reversal, inflammation, all the things that govern these neurodegenerative disease. But what we found was really the opposite. The actual efficacy or the effectiveness is dependent on lower amounts. So there's actually a curve that's opposite of what we would predict. The lower quantities give us the more significant outcomes. So we're in and I'm mentioning this, it's just early and it's in animal and laboratory assays, and we got a lot of work to do in this. But the idea that we can recover huge quantities from colostrum de novo, and we can use similar low levels that will elicit a better response, gives us hope that once we get this product kind of put together, we'll be able to know what the dose, the timing, and the utility ahead of what we're doing. And the fact, and I said this before, I'm going to say it again, but the fact that the colostrum is such a huge market with very limited clinical interventional studies is amazing. This is a multi-billion-dollar a year market with virtually no clinical intervention studies other than cosmetics. So this is a real opportunity to really start joining together and working together in something that's not only a product that we can consume for our diet, but also something that will, change the world or has a capability to make a huge impact as we look forward. Did people get that?
Yeah, can I be a cheerleader for you?
Yeah, well, you got to dress up a little bit. I mean, you got to.
Well, no, I'm not that bendy anymore.
It's all right. It's all right.
If I may.
You certainly may.
Thank you. He's a smart man to give it up. Hey, I just need to say you guys are so on the right track. I have been on an anti-inflammatory diet for 1.5 years, and I was diagnosed with MS 24 years ago. Since I've been on my anti-inflammatory diet, which includes the colostrum at Walmart, I haven't had any MS symptoms of flare-ups in over 1 year, and I would have those regularly. And so because I've gone all natural, and my aha moment was when it was on Channel 7 that you were doing this study. And I went, well, no wonder I'm not losing my right side. And no wonder my arthritis. I don't take any meds. And my arthritis was crippling arthritis.
Yeah.
And I'm still a chef.
Yeah.
And so food is my medicine. But you're on the right track. And if nobody here believes it, I can give you a lot of testimony to it. You are doing the right thing for
Yeah.
science.
Well, I appreciate that. I'd like you to come over Friday night and make dinner for us, though. I mean,
I'm not busy at Coach Roll's house, I'll be there.
Now, what differentiates the colostrum from Walmart from what you're doing? Like,
a lot. I think let me make it simple, and we can, as we get into the question and answer, Joel, I'm taking your time, so I don't want to devour that inevitably here. We're starting to dissect this in a very big way. No one has done it, to look at the colostrum and the products that we generated from colostrum. We believe that it is not only the active ingredient, but it will have a different mechanism or even a mechanism for how it works better. Now, I know we thought, and these lovely ladies are working on this. You better go back to the lab and get some more data, by the way. So I'm going to kill you. What we thought, and I'm going to tell you how crazy this experimental result was, and maybe Susmita will help fill me in here. You're still okay. We just thought that the molecules that are induced as part of the inflammation, and a big one, I'm just going to use a fancy word, it's called tumor necrosis factor alpha. It's a big inflammatory, you ever heard of that?
No.
Okay, so I'm going to say it's bad. It's bad. It induces, it's really bad. You don't want to take that home with you. It's really bad. It's part of the really big instigator of the inflammatory response. So we thought we can treat these cells with, the extracellular vesicles from colostrum or colostrum, and we can reduce the major inflammatory factor that really permeates every bad disease, whether it be MS, whether it be ALS, or Parkinson's, Alzheimer's, Huntington's, Alzheimer's, you name it, but it's a mediator of all this stuff. So we did these experiments. What did we find when we did these experiments with tumor necrosis factor alpha? What did we find? What? One sentence.
Okay, it reduced the TNF.
No, but there was no difference. What were the no difference? What is Dr. Gendelman saying no difference? There was no difference between what and what? Between what and what? Between colostrum and the active ingredient of colostrum. There was no difference. They were the same. And then we looked at something else, which was something that was very unexpected, that may shed some light on why this active ingredient of colostrum may be so much better. We still have ways to go, but maybe I'll let Dr. Sill comment on that one factor.
Yeah, so we looked into another factor which is called interleukin-10, and it is an anti-inflammatory cytokine or agent in our system. And with the colostrum, we saw there was a huge increase of this anti-inflammatory agent in the cells.
So what we're saying, and if it's a microphone?
No, it's just
Okay, I don't want any microphones. I'm dying with this microphone. I'm going to get a hearing aid. That's what I'm going to do, and it's going to solve my problem. We're all going to get hearing aids at the end of this. Forget about Alzheimer's and everything else. Hearing aids. But what was surprising, it wasn't the actual culprit. It was the one that regulates this culprit. It was the interleukin-10 is the one that controls these toxins. And we were looking at the toxin was saying, we got to find the toxin that's being affected. And we did see an effect, but it was very mild, very minimal. What really was happening is the factors that control this toxin production. So we were so excited about this. We were so excited about this that it really hurts me to say this, but I'm going to say it in public, that we gave these young students a time off to come to Elmwood, Nebraska when they should be working in the lab and getting these. Isn't that true? What? Yes. Terrible.
Because they're listening to all of this.
Barely. I tried. I took them out to dinner, to lunch. Did I take you to lunch, right? I didn't do that? And no, I'm sorry. I'll finish this. But we're all trying to find how, why, where, what's going on here, and how we can use this in the best way possible, right? Okay. I'm going to turn this over. I'll let you do the talking and me fall asleep a little bit. Go ahead.
Okay, last questions online. Are there other therapies like DDS? Sorry, can't read. Are there other therapies like DDS that would eliminate their ability to gain benefits from this therapy?
Do you know what DBS is? I'm sorry. Deep brain stimulation. Maybe I should get this. Yeah. I think the answer to this is that there's a whole bunch of different ways to skin a cat. But there's a big problem with all these ways, is that they only work for a certain period of time. Then they go away. They're amnestic. And what happens is if you take these medicines again and again and again, you either get toxicity, they become toxic, or they fail to work. That's why the natural way to go after this is absolutely the right way to go.
Yes, it is.
You agree with me?
I wholeheartedly agree. Remember, I'm your cheerleader.
That's okay, but
I know, you'd like somebody younger, but.
I didn't say. Oh my God! Help me! Oh my God! Help me! Okay. I'm going to erase that comment. It's not going to go anything going on. But yeah, it has to be. That's why we're really pushing. Whether it's a pharmaceutical or a nutraceutical, it has to be natural. Has to be natural based. It's very, very important.
Howard, let's go to Joel real quick, and then
Yeah,
we're gonna
yeah,
ask
yeah.
you questions after.
Yeah, that's good. That's good. Yeah, we're getting late.
Time.
Yeah, go ahead, ladies and gentlemen.
I'm going to be very, very brief.
Hey, go.
Joe, go. Yeah.
Wow. So I'm handling the business side of the company. And I just wanted to say,
Oh.
something that struck me. I hope you'll take this the right way. A lot of people talk about the rural communities as simple communities, as unsophisticated. I'll tell you two things about.
I'm stealing you for 2 seconds. Whether it's
Thank you.
online team.
All right, fair enough.
I'm not violating you.
Not in the least. Not yet. First of all, the hardest working people I ever employed were off the farm. No question, no close second, number one. Number two, I find it very powerful that diseases that occur naturally within the body may find the resolution in nature, in the nature that you work with. It's very, very powerful that the work that you're doing not only feeds America, but it may very well be that the work you're doing may end up curing or at least battling some of the most potent diseases in America. And to the extent that that's true, it's something to be incredibly proud of. So on the one hand, we drive out here and we see a lot of farmland and not a lot of buildings and all of that. But on the other hand, I see a lot of potential and a lot of character and a lot of strength. And so having been born and raised in this state, it makes me even more proud to be a part of this state, to know that the work we're doing through NeuroRegen may in fact be supplemented powerfully by the work that you do here. So I'm very grateful to all of you for that.
We really have to have individual questions and beyond what we did over the internet. And it's for you, Joel. And we're going to break a little bit to get something to eat but please, I implore you to talk to Joel, ask questions.
Well, I'm the one who's asking the questions, not so much them asking me, because you want for the interview. Right.
answer the question, but Joel will ask. Let name all around and.
I got a question for you that people might be wondering. Neural Regen has formed.
Yes.
How does this work?
Yeah, it's a legal entity registered in the state of Nebraska.
The hope is to go to human trials.
Correct.
We don't know when, but soon, we hope.
That's the aim.
Maybe you can outline just what it takes from this point forward to get us to human trials. If the general, I don't want to get you into trouble either.
You won't, because I won't allow myself to get into trouble. That's really more a question for Dr. Gendelman and his team. I'm basically here to facilitate whatever we need in order to make human trials happen, to make them successful, to present a good face to the public, an honest face to the public. But that's my job. Is the science stays with them, and I'm a facilitator. Whatever it is that they need to be successful, that's my job. Okay. All right. For the scientists, okay.
So I'm a little bit stuck on the mechanism of action here because so many neurodegenerative, there's a blood-brain barrier there for a reason, right? And it's impenetrable. So I guess I'm curious, how does this process that you're describing, this glycosylation, how does improve the permeability of the blood-brain barrier to allow to do things? I'm just kind of stuck on that because I work in biotech also, and we work in some epilepsies, and this is like the $1 million question in drug development is how to effectively penetrate the blood-brain barrier without causing any harm. So could you elaborate on that or hypothesize what?
Yeah, so the EVs by itself, they have a size from 50 to 200 nanometers. So they can easily cross the blood. So they can easily cross the blood-brain barrier. And when we are doing fucosylations, so they have the ability to attach to some receptors on the blood-brain barrier. Those are called the selectins. And so easily they can move through the blood-brain barrier. So it is actually, helping them to be more efficacious to navigate through the blood-brain barrier.
Can I ask a follow-up question?
Yeah.
So then the follow-up question would be, is there an example now in any other medications or studies that are currently ongoing where this method has been used and used successfully?
Yes.
Yeah.
Yeah.
Maybe not actually doing so the whole idea behind fucosylation is when we have this
Yours people like it here.
So when we have this cancer tissue, scientists found that there is a lot of
Just hold it.
So what scientists found that what clinicians found was that these tumors that are very hard for the immune cells to go places, the tumor also creates a wall around itself where it's stopping your immune system from attacking it. So one of the ways it does is these regulatory T cells, the same T cells that I said dampen the immune system, they attract more of these into the cancer tissue, and then they go really deep into the tissue. And when they isolated these regulatory T cells from cancer, they found that these cells are very highly fucosylated. So fucosylation acts as kind of a turbo boost for these cells to go further than where they're naturally able to go. So this idea was picked up and it became part of a company. Like we were working with Tagazyme, so the actual discoverer was someone else, but Tagazyme is the company that kind of tried to get it into the clinic. So basically the idea is if you fucosylate any cell type, this helps the cell go further than where it's supposed to be. It kind of lubricates the cell to go further. So coming back to your question about the immunoprivileged side of the brain, I mean, for a long time, people believe that the brain is immunoprivileged, but then T cells can go in and out of the brain, no problem. So our idea that we are working on, we have a $2 million grant from the Michael J. Fox Foundation, is that you take the regulatory T cells and fucosylate them and give it back to the patient. So now you have these turbocharged regulatory T cells that can go further than, so we're not necessarily guiding them where to go. It's just that they will go further than where they're supposed to go. So that way we can show, I mean, we already did a preliminary study in Parkinson's disease where we found that compared to unfucosylated T cells, fucosylated T cells have a much higher neuroprotective efficacy. So we're trying to expand that into the EVs too, where we hope that fucosylating the EVs will help them go further than where they're able to go. So that's the whole idea behind fucosylation of the EVs. But EVs by themselves is a big step up from just colostrum powder, but fucosylation hopefully will take it to the next level. So it's all, we have to work out and see if that's if the hypothesis works out in real life. Yeah. Oh.
All right, we've kept everybody quite a while. Is there any more questions online? Yeah. Well, so I'm going to leave that for afterwards. So I'm not going to step outside my lane here. He asked if if they're looking for investors or anything like that. I said, I'm going to leave that out. That's outside my purview. That's going to be afterwards here. So we'll talk about that. Let's break. I'm sure everyone needs a restroom break. We've been here for about 2 hours. There's good stuff. My wife cooked back here, baked back here. So let's get that. And then you guys can ask him that question personally. But thank you, Dr. Gundelman. Thank you to everybody who came. I hope it was enlightening. So, we are a very small part of this, but it's exciting. We're excited. So. Well, that's a short story, or long story. I'll make short. Keith Schwartz is related to my wife. They called. He used to work for the university. Said, "Hey, I know this guy. He's kind of crazy. Dude, you want to hear what he has to say?" That's a joke, by the way. He came down, sounded very interesting, and that's how we got started.
You have cattle.
We have cattle. It was luck of the draw.
Yeah.
So that's it.
You do.
Yeah. So thank you again, and enjoy.
Wow.